Hey, let's fucking go. So I got the audio figured out and everything should be working perfectly normal for this stream. So I'm gonna sit here and chill out for a minute. Um, we're gonna get into talking about genetic variables. We're gonna need to wait on my chat to pull up, so do not worry if you guys are contacting me in there. I will be able to respond momentarily, I hope. So, genetic, wow, off to a fucking great start in cannabis. Let's fucking go. Okay, so I did attempt to do this stream, so this is kind of a redo, but today we're going to hit genetic variables. I might release a video here, and then after I release another video, we'll probably end up doing another seed shopping stream, because I know you guys really like those, and I actually enjoy doing them quite a bit. I think they're very fun. Here, I'm going to see again if I can try to pull up this chat. I really don't like not having it up, but I guess we're going to have to deal. So, Cannabis Genetics studies the heritability traits and characteristics of the plant, determining its physical attributes, growth patterns, cannabinoid content, aroma, and flavor profiles. This study is crucial for developing new improvements in cannabis varieties through selective breeding. And when you talk about selective breeding, oh my gosh, there has been a whole lot of selective breeding. I might actually be able to pull it up over here. I don't know what the fuck I'm doing. I'll be able to pull up the chat and at least be able to talk to you guys until... Alright, there we go. Chat should be somewhere around here. I think it's on the side right here. Alright, but let's go right into genetic variables. So, understanding genes and plant breeding. I am over 21. Please leave me alone. Cannabis genetics involves studying their heritability, blah, blah, blah. We already talked about that. Phenotype and genotype, which, if you guys tuned into the last episode of Mr. Grode's podcast, we talked a little bit about pheno-hunting, which was really fun. I love being a part of the show, and having a conversation with him was super awesome. Phenotype refers to the observable physical characteristics of a cannabis plant, like height, leaf shape, flower structure, and resin production. Genotype, in contrast, is the genetic makeup of the plant, including specific combinations of genes responsible for its traits. Selective breeding aims to develop genotypes that consistently express desired phenotypic traits. So that is a very scientific way of putting, we're going to make things similar but different. Dominant and recessive traits. This is going to be new for me. I know some dominant and recessive traits when it comes to human biology, but plant biology is, I assume, quite a bit different. So dominant and recessive traits. Cannabis genetics involve dominant and recessive traits. Dominant traits are expressed even if the only gene copy is present. Only if one gene copy is present, while recessive traits require both gene copies for the expression. Breeders must understand these inheritance patterns to predict the traits offsprings will likely exhibit, guiding informed breeding decisions. So if you're trying to narrow that down and make an F1 or an F3 or 4 or 5 generation where you're narrowing down what you want as a genetic, you're going to be looking at the recessive versus the dominant traits, kind of doing the biological math there and knocking it out from there, making an informed decision of what you're going to have next. So organic cannabis cultivation. Organic methods enhance genetic expression, producing healthier plants that fully realize their genetic potential. Avoiding synthetic fertilizers and pesticides allow natural genetic traits to flourish. I can't put up too strong of an argument there. You guys know I'm a synthetic grower. That's primarily what I do, and I don't see a lot of problem with doing that. But yeah, having a natural thing where you're not doing a bunch of crazy stuff, the plant's able to do its own thing, that's all very good when it comes to making a genetic. History of cannabis hybrid strains. So getting right into the old strains, pioneering work began. You guys see this, right? They have a gray text on a white background. Could you make a harder article to read? Anyway, it's definitely not because I'm blind and need to see the eye doctor. That has nothing to do with it. Began in earnest during the 1960s and 1970s, a time where counterculture movement embraced cannabis and sought new ways to enjoy and cultivate it. Among the most pivotal developments was the creation of Skunk Number 1, which a lot of people are super familiar with. It's one of the more popular strains, I would say, at least the most popular strain. When you think of weed, you kind of think of Skunk Number 1 as a kind of default. A hybrid strain that forever altered the cannabis breeding landscape. Skunk Number 1 stands as a true testament to the power of selective breeding, having been developed to combine the robust growth patterns of Afghani indica strains with the potent psychoactive effects of Central American sativas, Colombian Gold. This strain wasn't just a hit due to its grow, ease. Wow. Holy shit. Wasn't just a huge hit due to its growing ease and strength. It set the standard for what a hybrid strain could be and opened up the door for a new era of crossbreeding, which is super cool. It kind of inspired the entire landscape of breeding and selective breeding and making a genetic, and that is super, super interesting. The importance of hybrid vigor cannot be overstated. The enhanced properties such as increased THC levels, which a lot of people aren't the biggest fans of, but some of them are it's for the right reasons. Improved resilience and yield, which is awesome. And it's something that I'm going to try introducing into my genetic when I start making them, is I want to have something that's highly resistant and very robust in its growth and also high in yield. Those are three things that I definitely know I'm going to be going for in that genetic. And then I have a general idea for terpene profile, but I still have to grow one of the parents out, get a little bit familiar, know what I'm dealing with on the other half of this thing, because it's super hypothetical. Advancements in cannabis genetics. The leaps and bounds in molecular genetics and DNA sequencing have provided us with a deeper insight into genetic makeup. One second. Hello? Uh-huh. Alright, I'm back, and I have water. Honestly, what the hell's going on? God damn it. Stepped on the cord and broke the light out of the wall. Alright, fantastic. Here we go. Alright, where were we? Leaps and bounds, genetic makeups, various strains. These technologies have been pivotal in identifying scientific genes makeups for traits such as THC and CBD content, resistance to pests, and growth patterns. Thanks to these scientific breakthroughs, we can now map cannabis genome with precision, which in turn allows breeders to develop strains with tailored characteristics. For instance, we now have a better access to high CBD strains that are becoming increasingly popular for their therapeutic properties, which is very cool. I actually need to grow out some CBD genetics. I really want to use CBD and take advantage of it and use it medicinally. I think it will be very positive, and I do recommend it to anyone out there who wants to get medicinal aspects from their plants. Because I think that's the easiest way to do it, especially if you're someone out there who's like, I want to try weed, but like I don't want to get high, man. Like, just go with the CBD strain, and then you'll get like this smoking experience. You'll know how it smokes, how it feels, how it tastes, without the intoxicating effects, which is a turnoff for some people. Let's face it. We can now map for instance, sort of targeting. This sort of targeted breeding was a mere pipe dream just a few decades ago, which is crazy to think about, that no one, like, before the 60s and 70s thought to breed genetics and crossbreed them and make something new. They were just like, man, let's stick with the landrace stuff. Weed is weed, man. It's very weird to me, and I'm sure there's someone out there who was breeding cannabis and crossbreeding it possibly in like the 18th century that we'll hear of in like 40, 50 years, and they'll be like, oh, that's where the duck foot actually came from. That comes from Hawaii. It's because someone went over there on like a boat with Columbus, and they found out that it's actually a cross between Afghani and Pakistani, and they sent it over there to trade with the native people. Some bullshit like that. I have no evidence to back up what I just said. It was literally a random tangent, by the way. Furthermore, the use of hydroponic systems for cannabis cultivation has seen a surge, primarily because of these systems that can sustainably boost the expression of a plant's genetic potential by providing optimal growing conditions without the variability found in soil gardening. Hydroponics is a game changer for the consistency and quality of your cannabis production. Breeders and growers can now yield harvests with maximized genetic expression, leading to higher quality and often more potent cannabis. To explore this further, we can delve. I've never fucking seen that word written. What the fuck? We can delve into the diverse hydroponic system for cannabis and their respective benefits. As our understanding deepens, we also recognize the value of genetic preservation and the use of seed banks. The repositories are essential in protecting the genetic diversity of cannabis and ensuring that rare and endangered strains aren't lost to time. They allow breeders to tap into a treasure trove of genetics, bringing forth unique combinations that would otherwise be inaccessible. These advantages have not just influenced the way breeders manipulate genetics, but have also transformed our approach to cultivation. I'm having a hard time. But have also transformed our approach to cultivation. With a growing emphasis on sustainability and efficiency, integrating advanced genetics into eco-friendly cultivation practices could be the next frontier in the cannabis industry. This synergy of cutting-edge science with traditional horticultural methods hints at an existing future for cannabis genetics and has profound impact on the strains we cultivate and enjoy. Very interesting stuff. The art and science of cannabis breeding. It doesn't go into saying which genetics are possibly dominant or recessive, which kind of... Hey man, you want to chill out? What's going on, Sage? What do you need? The art and science of cannabis breeding. Through our exploration of cannabis genetics, we've unveiled the intricate distance between nature and nurture. Our journey has shown us that the art of breeding is as much about scientific precision as it is about creative vision. We've seen how breeders sculpt strains to perfection, harnessing selective breeding and backcrossing. Pinpoint traits that meet specific needs. It's clear that the future of cannabis cultivation is bright with integration of the molecular genetics and sustainable practices, promising to elevate the industry to new heights. Let's continue to champion the preservation of genetics diversity as we step into an era where the full potential of cannabis genetics can be realized in harmony with our environment. A beautiful, beautiful way to leave that off. Fantastic. What role does organic... how might the future of cannabis cultivation integrate advanced genetics into eco-friendly practices? What significance did... Here, let's answer this question after I burp. What significance did the creation of Skunk Number 1 have on cannabis breeding? The creation of Skunk Number 1 represented a major advantage in cannabis breeding, marking the transition to hybrid strains. It introduced a precedent for developing strains with increased... Okay, they just kind of repeated what was in the article. Okay, no, they're just repeated answers, I suppose. Which is fine, I guess, if someone just wants to skip to that section and get the keynotes. I think it's fun to read the whole article. Backcrossing involves crossing a hybrid offspring with one of its parent plants to reinforce and stabilize a desired trait or desired traits. This technique is valuable for enhancing traits like THC content or specific terpene profiles. We've got selective breeding and open pollination, which I guess we skipped over. Selective breeding is a strategic approach where breeders hand select male and female plants to combine desirable traits. It involves identifying traits, selecting a parent plant, crossing, and stabilizing that trait across generations. This method customizes strains to specific needs, enhances traits like potency and disease resistant, and fosters innovation in cannabis genetics. Open pollination. Open pollination, a natural process where female plants are pollinated by male plants without human intervention, promotes genetic diversity, crucial for the resilient gene pool. It also helps in preserving landrace strains and enhances plant adaptability and stability. However, challenges include uncontrolled cross-pollination, leading to hybridization and variable offspring, making it difficult to maintain pure strains or specific genetic profiles, which I still think it would be super interesting to have an open pollination, but I don't know when I want to do it. Not that I would be having the males. I think I would just grow a field of females and hope that... hope is a weird word, but in that context, I would hope that they would get pollinated by another crop that someone else is growing that maybe hermed out, or they didn't know how to had a male in it, or they grew males on purpose and didn't give a shit. And I ended up getting all these seeds basically for free. Getting hundreds of thousands of seeds off a giant crop of plants. I think that would be really fun to see how those children turn out. Let's see if we can find another article. What year is this? Published year 2021, recent advances in cannabis sativa genomics. This could be the good stuff right here. And it's too bad okay, there's the summary. There's the introduction. This is gonna get fucking crazy. Holy shit, there's a lot of info here. There's a lot of really cool stuff. So I guess we should start at the introduction. Maybe something really cool will be in there. But I will be having to get off soon. Unfortunately, my family is going to be coming back home. So if I cut off the stream abruptly, that's what happened. Cannabis sativa. A member of the Cannabisaceae family is one of the oldest domesticated crops. Bradshaw, I'm not going to read anything in parentheses. You guys can if you would like to. It is believed to have originated in Central Asia, from where its cultivation rapidly spread throughout Asia and Europe. Nowadays, legal and illegal cannabis cultivation occurs globally. The exact number of species comprising the cannabis genus is controversial. Some claim the genus consists of 3 species that display distinct phenotypic differences, namely sativa, indica, and ruderalis. The alternative and perhaps most accepted viewpoint is that cannabis is a monotypic genus consisting of a single species, Cannabis sativa, which I don't necessarily agree with. I don't like when people call it Cannabis sativa. I understand it, and I'm not going to openly express it bothers me, but god damn it, this is my dojo. This is where I can be open and honest with the entire community. I don't like calling it Cannabis sativa as a general term. I've never liked that because we kind of integrated sativa as it being its own kind of geno, and then we have the indica and ruderalis. But not to get myself lost in bullshit, cannabis has a diploid genome 2N-20 consisting of 9 autosomes and a pair of sex chromosomes X and Y. It is predominantly dioecious, meaning a plant either is male or female, with estimated haploid genome sizes of 843 MB and 818 MB, which I'm not smart enough to know what those mean. For a male and female plants, respectively, despite the presence of defined sex chromosomes, environmental factors such as reduced photoperiod and low temperatures, and foliar applications of chemicals such as silver nitrate and the ethylene hormone inhibit thiosulfate-induced pollen production in male flowers, leading to the production of feminized seeds. This technique has been exploited as a useful tool in cannabis breeding. For example, selfing or crossing female plants, and in generating populations for dissection of the genetic base of important traits. That's dissection, I think. Is this just a long-winded introduction to cannabis as a whole? Because I just want to know what they found out. Alright, let's see if we can figure out this. Probably not, right? Got a bunch of chemicals here. They're moving places, doing some stuff. Interesting, I suppose. I'm going to try to zoom in here. That's too close. Okay, let's see if that'll work. Schematic diagram of cannabinoid biosynthesis. Biosynthesis including... Jesus Christ. Am I just going to get stuck on every other word here? Why can't science words be normal words? For the love of God, somebody. Polyketide and isopenide Presecure pathways are merged by a placid, localized, aromatic... What the fuck is that? Parenthiocyte? Jesus. God damn. Then they just hit me with another one. Reading this fucking article, to me, I suck at reading, by the way. I read at like a fucking 7th grade level or something. Reading articles like this is like fighting a fucking Elden Ring boss. I just can't do it. Wow. Acrylsacranic? I know that was wrong. Acids and geranyl diphosphate intermediates, forming cannabidioids with a linear isoperennial residue. Jesus fucking Christ. I'm not smart enough to understand this. Improve the prospects of using contemporary breeding techniques to improve cannabis traits with medicinal applications are promising. However, progress in this area is hampered by several issues. First, the genetics of cannabis is poorly understood and causes incorrect classification of cultivar/strains with implications for researchers, growing cannabis users, and regulators. Secondly, intensive clandestine breeding practices since the early 70s have led to a genetic bottleneck and reduction in allelic diversity in marijuana plants. Thirdly, restrictions such as the International Narcotics Conventions and associated legislations have hampered the exchange of cannabis genetics resources and research material, which is upsetting for everybody, scientists and users alike. I cannot wait until that gets changed. And I know a lot of other people are on the same boat. Cannabis sativa genomics resources and the discoveries they have enabled. Chromosomal nomenclatures of cannabis sativa genome assemblies highlighting Jesus Christ the discrepancies in chromosomes numbering among the current assemblies. The identification of the less studied CBCA synthesis is another notable discovery, or originating from genomics database. This gene catalysts the synthesis and cannabichromine, which is an energizing. Oh, that's CBC. So another cannabinoid, CBC, cannabichromine, which I'm probably saying wrong, which is an emerging target for medicinal cannabis breeders as it is non-intoxicating, can reduce pain sensations, and acts as an anti-inflammatory agent. CBCAS, THCAS, and CBDAS are highly similar to the notical and amino acid levels. The fact that long-read sequencing enabled assembly of such highly familiar loci reflects the importance of its technology and resolving complex repetitive regions in the cannabis genome. Our survey of the contig level PacBio genome assembles of pineapple, banana Bubba Kush, PBBK and Canatonic also support these findings. We identified a larger number of THCAS and CBCAS gene loci in these assemblies compared to ChemDog 91 and LA Confidential, which were generated using short read sequencing. It is likely that underestimation of THCAS and CBDAS and CBCAS loci has occurred in these collapsed and relatively similar assemblies. However, the presence of true biological variation among these cultivars cannot be ruled out. For instance, structural variants SVs in the form of copy number variations CNVS and presence abundance variations are known to affect the gene content of many plant species. Then we got these really cool charts. Man, this is a really interesting article. I'm going to actually leave it with you guys, the online library. I think this would be an interesting read, especially if I were smart enough to actually understand half the things that are going on. But I think I'm actually going to hop off for today. I appreciate you guys for joining me and learning about these variations in the genetics and kind of things that we're going to be looking forward to in the future, especially when it comes to more research coming out about these plants that we know and love. I do want to leave you guys off with the news that my Chill Out OG seeds that I started germinating on Sunday have germinated. 2 of them out of 3 have germinated, and I'm so excited to grow Chill Out OG. But for you guys who are new, please hit subscribe. If you guys like this live stream or any part of it, what's up, man? AB coming right at the end of the stream. Very happy to see you here. But yeah, Chill Out OG has germinated. I have a bag seed in there that I actually got out of my current crop. I was trimming some of my White Widows, and I ended up seeing 4 seeds that I found. So hopefully I can try to germinate those and see what actually comes from them and what I can get out of them and what may have happened to cause those, because I didn't see any. Nah, you're good, man. I just appreciate you being here and being in the chat. It really means a lot. I see you dropping those hundreds. I think that's you, at least. But yeah, no, I very tired, thinking about hopping off, but we did read a couple articles, so you can go back and watch those if you would like to. But leave a like if you liked it. I will see you guys next time. Let me know, actually, what I should do a video about, because I'm kind of running out of ideas for the time being. But I'll leave you guys with that. Thank you very much for joining.